The Animal Replacement Centre is a centre of excellence for replacing animals in research. It was set up to develop human models of human disease, which is the strapline that we've used for our research. And the aim very much has been to focus on developing human models of basal cell carcinoma of the skin and also human head and neck cancer. My opinion has always been that it's better to have a human model for a human disease. And we work in a laboratory that has developed and pioneered growing human skin cells. And so to us, it was obvious that the best way to investigate this cancer, to understand what it's doing in human cells, is to actually mutate the pathway in human cells rather than a mouse.
The Animal Replacement Centre is designed to focus on areas where animal models are widely used, and then to provide replacements for that. So for example, tumor invasion, tumor metastasis. And so what we do here in the center is we try to model that in 3D. We try to develop new techniques as well to be able to one day offer to the scientific community a set of resources that can replace the use of animal models.
What we're doing now in the Animal Replacement Centre is improving our models so that they're more human relevant. So rather than just using these 2D dish models, we are doing things where we're growing cells in a 3D matrix that mimics the environment inside a person. We're also taking actual tumour specimens out of people, so rather than these cancer cells which are growing in a dish, we're taking some tumour specimens directly from people. And we're doing two things with those. One thing is we're growing those in a dish, so they get in 3D. And the other thing we're doing is taking human tumour specimens that have been preserved and looking at those, so you actually get the architecture of the original tumour and comparing that to the types of cells we're seeing in our cell lines. The idea is by bringing together all these different animal replacement models, we'll have a really strong body of work. Of mice in cancer research particularly, and that's the main animal we're replacing. What we want to do is generate models which are actually showing human physiology. They are mimicking the human environment. And the main advantages of animal replacement research are firstly that it's much more reproducible than animal research, and also much more controllable. You can control the variables like you can't do in an animal, and you can also take very detailed measurements, again in a way that you can't really do in animals. So it's exactly the sort of technique that we need to be able to develop in order to replace animal models, because this is going straight from the human environment into the human-like environment in vitro.
We're just leaving all the mouse behind now. That's like an old technology. We're moving ahead now, and we're doing it so much faster, and it's all controlled. The data is much more clear and more valuable. And we can do many experiments with 100 drugs, whereas if you want to test 100 drugs, you have to sacrifice 100 of these mice.
The reason we use human models is basal cell carcinoma is a human skin cancer. Human skin is very different to mouse skin, and so mouse skin is very thick. It's covered in fur, it doesn't have any sweat glands. The way it divides is different. It divides much quicker than human cells, human skin, and traditionally mouse skin is very quick to transform and form tumours, whereas human cells do not want to form a tumour, so it takes many more mutations to cause a human cancer than it would a mouse cancer. And so we just think that using human cells, when we have those human cells, is the appropriate way to understand that cancer.